Welcome. I'm happy to present our research on mechanisms of metachronal coordination and explain why, in the presence of biological noise, synchronization of cilia is local but not global anymore. Many multiciliated epithelia have cilia that beat in a coordinated fashion with a fixed phase relationship in the form of a metachronal wave, similar to a Mexican wave that travels through a soccer stadium, which allows to pump fluid efficiently, for example, in our airways. So what synchronizes cilia? It has been hypothesized already by Sir Taylor that hydrodynamic interactions between neighboring cilia can synchronize their regular beat. In a nutshell, when a cilium beats, it sets the surrounding fluid in motion, which impacts hydrodynamic forces on neighboring cilia, which slows down or speeds up their beat and establishes a fixed relationship between neighboring cilia. But it was only recently that this hypothesis was experimentally proven. However, one question prevails, namely, are such hydrodynamic interactions between cilia strong enough to overcome the adverse effect of active fluctuations? Because each beating cilium is a noisy oscillator displaying frequency jitter as a consequence of the stochastic action of molecular motors inside. And to address this question, whether we still find metachronal coordination if we account for this biological noise, we developed a computational model. Now, this is not the first computational model. There's a rich literature of minimal models, for example, analyzing the cilia beat as the motion of a sphere moving along a circular orbit, which give a lot of important qualitative insights but have limited predictive power. On the other side, there are detailed simulations that run on large supercomputers that really aim to be quantitatively accurate. We try to bridge these two worlds with a multi-scale modeling approach where we use cilia beat patterns from experiments, but simplified interaction logic. So here's how it goes. We take a three-dimensional beat pattern from a unicellular paramecium, a multicellular algae, and this beat pattern was still traced by hand by Mechimer, and we plug this 3D beat pattern into our hydrodynamic simulations. We compute the forces exerted by the cilium on the surrounding fluid and the flow patterns, and with this we can accurately determine the hydrodynamic interactions between neighboring cilia, and then scale this up to an entire cilia carpet where each beating cilium follows the sequence of shapes of the paramecium beat pattern, but the speed, how fast each individual cilium beats, depends on the hydrodynamic interaction with its neighbors. And we can then study the emergence of synchronization and metachronal coordination. There's a whole zoo of different wave modes that's possible. This is what physics tells us. Of course, the most simple is that every cilium would do the same if they all beat in unisono, and this corresponds to so-called in-phase synchronization. But there are other wave modes. For example, a traveling wave where cilia beat with a fixed phase relationship, giving rise to a traveling wave with a well-defined direction and wavelengths. And for each possible wavelength, we can compute, using our model, whether this wave mode would be stable to small perturbations, because in an experiment, we will only be able to observe such stable wave modes. And here, every wave mode marked in green is indeed stable. These are still quite a number of different wave modes. So we can ask which wave mode would actually be selected by nature. Well, for this, we have to study what would happen if we start with random initial conditions. And then we find that although there are multiple different wave modes, all of which are stable to small perturbations, if we start with random initial conditions, they will predominantly converge to one dominant wave mode. depicted here. And this happens to be a daxioplectic wave where the wave direction is rotated counterclockwise to the direction of the effective stroke of the beating cilia. And indeed, in the model system Paramecium, from which we took the 3D beat pattern for our simulations, cilia synchronized as such a daxioplectic wave. So this was still without noise. What happens if we now switch on active fluctuations of the cilia beat if we account for the known frequency jitter? In that case, we find that locally cilia still synchronize, but on a large scale, say on a tissue scale, there are only local synchronized patches, and there's no global synchronization anymore. However, such local synchronization is probably all that's needed, because if we compute rates of fluid pumping, we find that local but not global synchronization already gives almost maximal rates of fluid pumping. So that's sufficient for the physiological role of fluid pumping of a cilia carpet. Now, that has been all theory. Can we test this in experiment? And for this, we are collaborating with the Jurisch-Yaksi lab in Norway, and they are studying the ciliated nose pit of zebrafish, which is a highly accessible model system to study the dynamics, self-organized dynamics of cilia in a cilia carpet. Many physiological parameters are very similar, in fact, to those of airway cilia carpets. However, this model system is much more accessible. And together we developed the algorithm to compute the phase of beating cilia, here depicted in rainbow colors. And moreover, we can compute for every pixel in this movie how synchronized it is with neighboring pixels. Is it oscillating with a fixed phase relationship with neighboring pixels? And if we do so, we find islands, local patches of local synchronization, where indeed cilia beat with a fixed phase relationship, but there's no global synchronization. But this local synchronization is sufficient to still give rise to these consistent and persistent metachronal wave patterns. And with this, I want to thank you for your attention.